Okay, good afternoon. So yeah, we'll be talking about plants, trying to concentrate on some aspects that are peculiar probably for plants. And but before doing so, talking about gene editing in plants and the legislation in Europe and also comparing it with the legislation in other parts of the world, I would like to start by putting a little bit of context. And the context in plants is plant breeding. So I think that this one. Sorry for that. Okay, sorry for that. So I'm putting this because I think it's important to understand that when we now talk about editing or modifying the genome of plants, and this will also apply for farm animals actually, we have to take into account that humans have been modifying the genome of plants since the Neolithic times, and they have done it through history, applying at each moment all the techniques and the scientific knowledge that was available at that time. And this means that this process, in which we see here at the bottom transgenic plants and now gene-edited plants, has been done with an increasingly scientific basis, but also an increasingly technification. So the process is more and more technically complex. This process has completely changed the genome of these plants in characters, in traits that are essential for living in the wild, and that are also essential for changing the type of products that we want to obtain from these plants. So here there are a number of these traits, but there are many of them. And this means that at the end, we have completely changed things like, for example, the architecture of the plant. Here you can see maize and its wild relative teosinte, or the cobs, which are completely different, or as we were discussing this morning, the wild tomatoes that were this size, or the present tomatoes that we cultivate that are this size, apart from changes in the composition that are quite important. So humans have profoundly modified crops and the products that we obtain from crops, and we can even consider that plants that we grow are actually designed by humans. Okay, so when we talk about natural, what is natural and what is not, this is a very misleading word where we put a lot of different concepts behind and not always clear what we are talking about. So what has happened during all these times is essentially we have been increasing the variability that we can use for breeding, starting from the same species, locally grown, that could be crossed naturally, to essentially any species with transgenesis, because we do not rely on crossing. And also, not only using the existing variability, but also inducing new variability, for example, with conventional mutagenesis that has been applied to breed plants since the 50s of the last century. So it's quite a long time that we are using radiation, X-rays or gamma rays, or chemicals to induce more mutations and be able then to select from this variability what we really want to improve. And now gene editing is the new technique that we add to this collection, or this toolkit that breeders have on hand to improve plants. So what is the legal framework? And I will be talking essentially in the EU, but with some considerations also on the framework in other parts of the world. So for most of the new varieties that are being developed, there is only a registry of varieties of the different member states that is somehow coordinated at the European level, where you have to demonstrate that the new variety is distinct, novel, it's homogeneous and stable, but there is no need for any risk assessment of these varieties. And I think that it's important that we remind that there is no such a thing as zero risk when you talk about food. There is plenty of different food that we eat every day that have a certain level of risk, in particular for some groups of the population. Okay, so there is no need for risk assessment. In case of GMOs, there is a completely different legal framework. Here, risk assessment is at the core of the legislation. So I'm just going to go very briefly on the European legislation, just to show you that what is a GMO in legal terms is not necessarily what you will consider a GMO as a scientist. It is very clearly defined, in the legislation, and a GMO is any organism, with the exception of human being, in which the genetic material has been altered in a way that does not occur naturally. Okay, and naturally, again, this word that is a word that we should try to not to use because it's very misleading, has to be defined. What do we mean by natural in the legislation? And it says in the terms of this definition, genetic modification occurs through the use of the techniques that are listed in one particular page and one particular part of the legislation and in an annex. And other techniques that are listed in another annex do not give rise to a GMO. So clearly we are talking here about techniques and not really the product that we are obtaining. And these techniques are any technique that uses recombinant nucleic acids, and new combinations of genetic material, again, in a way that does not occur naturally, which is... yeah, sorry. And what is also interesting is that this legislation also says that there are some techniques that give rise to something that we can call a GMO, but they are excluded from the regulation because the reason for that is that there is some history of safe use of these techniques. And this is chemical and radiation mutagenesis, which is not considered, which is not regulated in Europe. Okay? So clearly we have here a legislation that is based on the process, which is different to what happened in other parts of the world, which essentially what is regulated is the product. If the product is different, then it has to be regulated. If you can obtain the same product by different techniques, they are supposed to be regulated in the same way. This is not the case in Europe, although there is, or there was, I should say, some consideration to the final product because the approach was case by case to some extent. This, I have to say, that has been essentially lost in the last years because now there is a binding regulation that requires every single application to follow exactly the same approaches. So just very quickly, this is the process where the European Food Safety Authority has a key role in assessing the risk assessment performed by the applicant. And what is specific in Europe also is that there is a clear distinction between risk assessment process, the risk assessment part, which is supposed to be essentially scientific, and the risk management that is completely political. In other parts of the world, risk assessment, the competent authority that performs the risk assessment is the same that then manages the risk. Here it's completely separated. And the idea was to really increase the trust of the public in the sense that the risk assessment should be done by a completely separated agency that was only scientific, but the reality is that the risk managers really influence also or try to influence this assessment. So this is when these dossiers were on paper. Now they are no longer on paper, thanks God. And this is the size of the amount of data that you need to provide. This costs around 10 million euros, and the process can last for 5 to 10 years, and in some cases it never ends. So, for example, all the applications for cultivation since '98 have never been approved. So, what about gene editing? In legal terms, there is the different uses of gene editing are divided in 3 different categories. I will not be talking about the use of nucleases or CRISPR, for example, to introduce transgenes at a particular location, because this is essentially a transgene. This one is when you use CRISPR or any nuclease to cut, and then introduce random mutations at a particular location, and SDN2 is when you introduce a particular mutation at this particular locus. So what are the safety problems or that the regulation should look at? The intended modification, and actually nucleases or CRISPR will introduce changes that are indistinguishable from mutations that are happening spontaneously or that are introduced by conventional mutagenesis, and therefore the risks should be similar. This is in principle what you would expect. For the non-target effects, and we have heard before today that there are ways of limiting these off-target effects. I think that we need to take into account that the background of spontaneous mutation for every generation in a wheat plant, for example, it's around 200 new mutations. In genomes, in smaller genomes like tomato, it will be like around 10. When you do mutagenesis, you usually introduce around 600 mutations. So this is also a background that we need to take into account when we talk about off-targets. But also, in the case of plants, as for traditional plant breeding, non-target modifications can be segregated away in most of the cases. So what is peculiar or what is different when you work with plants is that you produce a lot of different individuals and then you select the one that you want. And then you usually backcross this new mutation into lines that are well adapted for growing. Therefore, you have a number of crosses in which you select each time your modification, and therefore the rest of them, if they are not linked genetically, they will be lost. So what happens in different parts of the world? So since quite a long time now, the USDA has been treating these genome-edited plants where there is no genetic material. For example, the CRISPR machinery, in this case this was in finger, are not integrated in the, or are no longer present in the plant. They will not be regulated. And in the case of more important changes, like what we call SDN2, we will consider case by case, but in principle, if there is no transgene present, they will not be considered this as GM. And we have already heard that there is already a product on the market. This is the soybean obtained by Calix, in which they have changed the oil composition to obtain a healthier oil. This is already in the market in the US. And there is a number of products that are very close to commercialization and that USDA has already said that they will not regulate them. Okay. What happens in Europe? Europe is more complex. This is in UK. The field trials in Europe are the responsibility for the oversee of the field trials is at the national level, and there are different competent authorities of different European member states that have considered these plants as non-GM, and this is a field trial in the UK. But when you want to commercialize this belongs to the EU, the responsibility, and EU, it's a different story. So in EU, there has been scientific consensus and some, as I said, some national advisory bodies or competent authorities that have already issued documents considering that these should not be considered as GM. There have been discussion among member states since 2008. The European Commission set up a working group with different competent authorities of the different member states that have been discussing for 10 years with no consensus. And last year there was this ruling that was triggered by some questions from the French Conseil d'État that sent to the European Court of Justice a number of questions very clearly asking, okay, considering the present legislation, should we consider gene-edited plants as GMOs or not? And you all know that the answer from the European Court of Justice that was published 1 year ago was clearly saying they have to be considered as GM. This is an interpretation of the law of 2001, and the only thing that the court did was to interpret this law and try to put gene editing that did not exist, obviously, at that time, into this law that is quite old. So what is the situation now? The situation is that the law requires any GMO commercialized to be detectable. So you need a specific detection method for any GM, which is something that you just cannot do for a genome-edited plant. And the thing is that the detection and the traceability is responsibility of the member states. It's not at the European level, but at the member states. So all member states, all competent authorities in Europe are quite nervous now because they have to do something that they cannot do. So this indeed open now a discussion that is not finished yet in order to know how are we going to deal with this. Obviously, there are other consequences which are very clear. So only big companies or trades that have a high return will be edited in Europe, because small companies cannot afford this investment. And obviously then the European seed companies will be left behind of the companies from abroad, which is exactly what has happened for in the case of GMOs. And the European food sector will not benefit from the advantage of this technology. I have to say that for GMs, in Europe we are only growing one GM that was approved in '98, which is a maize resistant to BT, to a pest. But we are importing hundreds of, more than 100 different GMs, because Europe cannot produce enough maize and soya to feed our animals, and therefore we are importing these, and the only consequences is that our growers cannot grow GM. Okay. So because of that, there have been quite a lot of debate during this last year, and actually even the group of chief scientists of the European Commission has produced a document in which they say that something needs to be done, the legislation needs to be revised. And actually there are essentially two different possibilities now. One is to reintroduce flexibility to the system in order to reduce data requirements on a case-by-case basis and treat genome-edited plants in a way, even if it's in the same process, but in a way that is different somehow. For example, not requiring a detection method, or reducing the number of requirements, or just changing the GMO legislation. For example, one of the proposals is to include gene editing in the list of GM, but not regulated like conventional mutagenesis. Well, all this is open. The discussions are ongoing, and we will see what happens when the new commission starts to work. Okay. That's it. Thank you.
Thank you.